Hi, I'm Jennifer Belvo. I'm one of the lab instructors for BSc 2010L, and today we're going to be working on Lab 6-2, isolation of bacterial DNA. For our experiment today, we will be using 6 microcentrifuge tubes total. So before we start doing anything with our E. coli, we're actually going to go ahead and label our microcentrifuge tubes ahead of time with the appropriate labels. So 3 of these microcentrifuge tubes will be holding our vector bacteria, and the other 3 will actually be holding our recombinant plasmids. So we're going to go ahead and label V1, V2, V3, and R1, R2, R3, along with our initials on each of these microcentrifuge tubes. For this lab, we're going to be working with sterilized equipment. So I just wanted to point out a few pieces of information that will be evidence to let you know whether or not something like our micropipette tips are actually sterilized or not. So with each of our steps, we will be using, of course, our gloved hands to keep ourselves safe, but also in order to prevent DNA contamination from our DNA getting mixed in with our bacterial DNA. With our micropipette tips that we have at our table, one thing we'll notice is that on the top of each of the micropipette tip boxes, we'll see a piece of tape that has some slanted dark lines across it. And this is actually what's called autoclave tape. And so what happens for the sterilization process, we have our tools or our items that are being used that get placed into an autoclave for sterilization purposes. Prior to the sterilization process, in other words, prior to being placed into the autoclave, items will often have autoclave tape placed on top of it. When this autoclave tape is first placed on top of the item, it's actually white in color. It's the actual process of going through the autoclave process that reveals these black lines on the tape. And that's how we know that this particular box of pipette tips has actually been through the autoclave, is because my autoclave tape is now showing those black lines on top. So that's one little piece of information to know about our sterilization process when we're working on this lab. Now that I have my sterilized pipette tips, I'm going to go ahead and get my micropipette. And the first step is to actually pipette 1.4 milliliters of each different type of E. coli bacteria colony that I have. So I'm first going to pipette 1.4 microliters of our vector bacteria into each of 2 tubes, microcentrifuge tubes, and then I will pipette 1.4 microliters of our recombinant plasmid into each of the 2 tubes that I have labeled with the R. I have my P1000 set to 700 microliters, and what I'm going to do is at this setting I'll pipette twice in order to pipette my 1.4 microliters. So I will pipette 1.4 microliters into my tube I have labeled V1. And into tube V2, I will once again pipette my 1.4 microliters. It's very important to change our pipette tips between our samples so we avoid cross-contamination. So I'm going to go ahead and inject my tip into the appropriate waste container. Since this is considered hazardous waste, we'll dispose of this in our hazardous waste bin, so that bin can actually be autoclaved prior to discarding it into the regular trash. And similar to my pipetting that I did with my vector bacteria, I'm going to go ahead and pipette 1.4 milliliters of my recombinant plasmid into 2 of my microcentrifuge tubes that I have labeled R. So I have since changed my pipette tip. So this is a brand new micro pipette tip. One. So there are 2 microcentrifuge tubes labeled V and R, each containing 1.4 milliliters of our 2 different E. coli bacterial colonies. To operate our microcentrifuge, we need to first open our lid. We're going to pull up on our lid lock here in order to remove our lid. And when we spin our samples, for our first spin, we're going to make sure that, first of all, we're going to make sure that our hinges are facing out and that our samples are balanced within our centrifuge. So we want to set them across from each other. And I'm going to count the number of holes between each side to make sure. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 10 on each side means we're balanced. And our lid is going to go on, get locked in place. The lid gets closed. For our first spin, we're going to just be spinning for 2 minutes. So our timer dial is on the left-hand side. So by moving this dial, we can see that the number of minutes will increase. So I'm going to set it to, that's 1 minute, that's 2 minutes there. And we need a speed setting, which our RPMs are set on this, with this dial on the right-hand side. And we follow the top graduation there. So that's at 5. And so all I do then is hit start, the start button, and we'll start spinning. After removing our samples from their first spin in the microcentrifuge, where we spun our samples at an RPM setting of 5,000 RPMs for 2 minutes, this first stage left us with a beautiful separation of our media that we actually grow the bacteria in and the actual bacteria cells themselves. So if you look at the bottom of our microcentrifuge tube, we actually now have our supernatant, which is our media, and in the very bottom we have a clouded area that's actually the collection of our bacterial cells. So this first step is actually for the purpose of separating our bacterial cells from our media that they actually grow and reproduce in. So at the first stage, we have our supernatant and our bacterial cells that have formed the pellet following our first round of centrifuging. So we're going to go ahead and dump off our supernatant since we don't need that anymore. So we are going to pour our supernatant off, and our pellet, which contains our bacterial cells, will actually stick to the location at the bottom of the centrifuge tube. Now we do have some extra media that has stayed in place because of surface tension. So what we'll do is take a P200, we're going to set it to 80 microliters, and we're actually going to manually remove that excess supernatant that's hiding underneath our microcentrifuge tube. Now the reason that we actually put our microcentrifuge tube hinge out is so that we always know where that pellet is going to form. So with our hinge located here, we know that the pellet will always form at the top side of the microcentrifuge tube. What this allows us to do is go in underneath our pellet and actually remove our excess fluid. So we're going to go ahead and suck out some excess fluid. from underneath our pellet. And we have left behind our bacterial cells in our microcentrifuge tube. So we're going to go ahead and do the same thing with our remaining 3 tubes in order to have just our bacterial cells sitting at the bottom of our centrifuge tubes. The next step in our process is to add 70 microliters of stet buffer into each of our microcentrifuge tubes. So we're going to find our P200 and set our P200 to 70 microliters. And our stet buffer is ready. So each of our centrifuge tubes, which right now I have sitting in a little ice bath, we're going to go ahead and add 70 microliters of stet buffer to each of our tubes. In order to avoid contamination, of course, between each addition, I am going to change my pipette tip. Here's our V. And our last sample. Once we've added our stet buffer, we're going to go ahead and vortex each of these microcentrifuge tubes in order to get our bacterial cells back into suspension. So right now they're still as a pellet at the bottom of our centrifuge tubes. We're going to vortex our microcentrifuge tube in order to get that pellet so that it goes back into floating around inside of our stet buffer. So we're going to vortex each of our samples, something we've done several times now. So I'll go ahead and vortex all of my samples off camera, but just to show you that we now have our stet buffer, which was clear, now has a bit of a cloudy look to it, and that's simply because we have our bacterial cells now floating around in our stet buffer.